Ingamana ingareo, ingata widapumanawa, ro rakatidama, tenakoto, tenatato katoa. Good evening. My name is Harleen Hain, and I have the great privilege of being the Vice Chancellor here at the University of Otago. And it is my great pleasure to welcome you on behalf of the University to this inaugural professorial lecture for Peter Dearden. Now, I'd like to extend a particularly warm welcome to the members of the wider community who are joining us this evening, and including Peter's family, who I suspect are quite rightly here with a huge sense of pride. In particular, I'd like to acknowledge his wife, Petra, his 3 children, Victoria, William, and Elizabeth, his father, Emeritus Professor Chris Dearden, and his mother, Monica. Now, like all of our promotions, Peter's promotion has come on the back of years and years of hard work and scholarship. In order to achieve promotion to this rank, an individual must demonstrate excellence in teaching, in research, and in service to the university and the wider community. And Peter is absolutely no exception to this rule. He has certainly excelled across all 3 of these categories. In terms of research, Peter has an impressive publication history on developmental plasticity and epigenetics. His work on honeybees in particular has been supported by a long string of extramural research grants and for work at the interface with industry. In terms of teaching, Peter is a popular classroom teacher and a popular research supervisor, and I understand that a large number of his research contingent are here this evening to support him. But among all his strengths, perhaps Peter's greatest strength is his ability to translate complex science into language that captures the imagination of the general public. Over the years, he has led a number of highly successful community projects through his work at Genetics at Otago, Gravita, Hands-on Science, and most recently with Lab in a Box. His efforts in this area have been rewarded with the Callahan Medal for Excellence in Science Communication from the Royal Society of New Zealand. But for me personally, Peter's greatest strength is his enthusiasm for everything that he does. Now, although good attitude is not a promotion criterion, although perhaps it should be, Peter's generous and affable manner means that working with him is always a pleasure. It also means that when he pushes the boundaries, which he does frequently in his career, the university is more likely to turn a blind eye and allow him to carry on. Now, we've just completed the professorial promotion round for 2017, and it was a poignant reminder for me about exactly how difficult it is to be promoted to professor here at Otago. Now, not only do we apply our own extremely high standards, but we also seek the views of internationally recognized experts in the candidate's area of research expertise. Now, when we sought this kind of input regarding the merits of Peter's promotion, we heard things like, the first time I heard him speak at an academic conference, I was stunned by the quality of the research presentation I heard that day, both in terms of the elegance of the Drosophila and honeybee research presented and the eloquence of the speaker. We also heard, Peter Dearden is an extraordinary academic and is most worthy of the promotion. Simply put, Peter is an outstanding and creative scientist who is without doubt a leader in his research field. And finally, the University of Otago is very fortunate to have him on the faculty. Well, Peter, I couldn't agree more. I've always been happy to have you as a colleague, and I would like to congratulate you on behalf of the wider university for your very well-earned promotion to professor. I will now call on Professor Vernon Ward, who is the Dean of the School of Biomedical Sciences, to tell us a little bit more about Peter's journey to professor. Nō reira, tēnā koutou, tēnā tātou katoa.
Thank you, Vice-Chancellor. Tēnā koutou katoa, hame nui tēnā kia koutou. Welcome, everybody. I have the very pleasurable task of introducing Professor Peter Dearden to you this evening. And getting to introduce people for the inaugural professorial lecture is one of the pleasures of the job, and it's always nice to watch their reaction as they sit and wonder what I'm actually going to introduce. Always a little bit nervous for them. So one of the good things I get to do is to look at the journey somebody has taken to get to be professor, and it's always a fascinating journey. Peter's interests have been in the area of genetics pretty much from his early days. He undertook his undergraduate and honours degree at Victoria University in the early 1990s, and then he moved on to undertake his PhD at Imperial College in London. He followed that with 2 postdoctoral positions in zoology departments, one in Cambridge University and one at the University of Western Ontario. He then came to Otago in 2001 as a research fellow and very quickly moved on to be a lecturer, senior lecturer, associate professor, and 15 years later, he is now a professor at this university and very deserved along the way. Throughout that time, Peter has maintained his interest in all things genetics, especially as that relates to evolution of the animal form. One area that I'm also going to emphasise, following on from the Vice-Chancellor's comments, is that it's very fitting tonight to recognise Peter's commitment to science leadership and his commitment to science communication. And I was trying to think of what's the best way of illustrating this, and so I thought I'd just read a few of the awards and recognitions that Peter has received during his journey. The Callaghan Medal, as already mentioned, the Ross Crozier Medal from the Genetic Society of Australia. He's been Deputy Director of Gravita, a Centre of Research Excellence. He's on the Otago Museum Trust Board. He's been Vice President of the Otago Institute. He's the Director of Genetics Otago, which has just been renewed. He's on the Ministry for Business, Innovation, Employment, Science and Society Reference Group, the Science Media Centre Advisory Board for the Royal Society of New Zealand. He's won the Rohith Trust and Carl Smith Medal in this university, the inaugural convener of the Ozone Group, and perhaps most interestingly, I think, is the Lab in a Box. It's one I like. I think it's brilliant. And I could keep on going for at least that long again. And then I sit down and saying, well, how do I also show what Peter's commitment to communication has been? And I added up and looked at what he's done this year. 46 media presentations, commentaries, articles in the popular press, on radio, TV, that sort of thing. He's genuinely committed to it. Of course, to be a professor, you have to have some decent research to underpin the communication, and he does. He's got 66 publications in journals such as Nature, Nature Communications, PNAS, and I note, quite fittingly, a lot of his publications have come out in things like the Public Library of Science and Biomed Central stables of journals, which open access fits very much with what Peter's emphasis is on. He's been funded by Marsden, MB, CORS, HRCs, international funds, and many other places as well. I haven't covered his teaching. It's stellar, and you're about to see that. And that's my segue to saying what you're going to hear tonight is his presentation on shaping Darwin's endless forms most wonderful. What you'll see is Peter's career-long interest in genetics, his fascination with how genes and processes influence the evolution of animals and how this world has ended up with such a broad, wide and diverse collection of living entities. That's his interest. What he also does is he goes on and takes fundamental knowledge and applies it, whether that's getting a better bee or whether that's the controlling of the pesky insect. That's his areas of interest, and he applies it. So there's a no-pressure build-up there, Peter. I'd now like to invite you to come forward and present your inaugural professorial lecture. Nā reira, tēnā te mihi kia koutou katoa. Tēnā koutou, tēnā koutou, tēnā tātou katoa.
Well, hi. Hello. I was sitting there thinking, this guy sounds good. It's going to be an awesome talk. And then, disappointingly, it's me. Hello. My name's Peter. Thank you so much for such a eloquent and mainly true introduction. It's very kind of you. It's always nice to have people say nice things about you. Yeah, actually, no, maybe it's difficult. And I'd just like to thank. That's very kind of you, and thanks for the professorship. That's very I appreciate that. I'd like to say thanks for my family for coming. Hi, family, folks. Are you guys going to be good? Good. Nice to hear. Okay, and I'd also like to thank, because it's not really on a slide, but I'd like to thank the Biochemistry department, because you guys have made a home for me. And I remember John Cutfield, ex-head of department, saying, you know, one of the things that we like about Peter is he's not a traditional biochemist. And I think I muttered under my breath when I heard that, not a biochemist at all. So thanks very much for having a cuckoo in the nest. as it were. Oh, there's so many people here. Okay, the title today is from Charles Darwin, and I was going to just call it Endless Forms, but then I thought you might not come because it'll be about biological compliance. It's the quality of the joke we're going to get tonight. Doesn't get any better than that. So, Charles Darwin wrote a book, I'm sure you've read it, The Origin of Species, and you don't need to read it because the only bit that's worth reading is the last paragraph. Every other paragraph in the book is a lead up to the last paragraph, and this is a bit from that where he says, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. And this has been a question for me, is that endless forms thing. How do we produce the forms of animals, the shapes of animals? How are these things put together? How are they controlled? How does that relate to the genes and the pathways that make an animal, and how do they change? And that's what I want to talk to you about today. Up there's some pictures of beetles, right? And beetles are cool. In fact, beetles are the most speciose group of animals on Earth. They're far more interesting than we are. But there's just a selection of some beetles, and you can look at them and say, actually, this commonality of form, some of them are different colours, isn't it pretty? What a nice diagram. And that's what Darwin was talking about. He was saying for the first time, really, that these things all descend from a common ancestor, and they're all modifications of a basic pattern. I think that's a really interesting idea. The problem comes when we take that information and we project it to this picture of diversity. So this is a picture of all animal phyla on Earth, I think. I may have missed some of the really weird squishy ones that live between sand grains. But while it's easy to see how we get between those different forms, when we're talking about beetles, how do we get between the different forms of these remarkable and out of focus on this side animals, right? Yeah, much better. So up there are two important ones that you should probably care about. There's a mouse up there, and there's an insect, right? Those are remarkably different animals. There's a lot that's different about them. The very body plan is different. And that's what we mean when we talk about a phylum. A phylum is the level of classification just below the animal kingdom. These are the big, different sorts of groups of animals. How do we get from those? How did evolution shape the body forms of whatever the ancestral animal looked like to produce all of that different diversity? Because that's the diversity that really underpins animal life, right? Beetles are all beetles. But actually, there's a remarkable diversity just there. So when I was an undergraduate student at some northern university we won't mention, Jeff Rickards, a famous or infamous genetics lecturer, we used to call him Ming the Merciless because he had one of those beards, he set an essay question when I was an honours student, which was a typical Ming the Merciless kind of question. It was just, does macroevolution equal lots of microevolution? Are the processes that made those little differences between those beetles the same processes that made those differences? And I suspect he'd been setting that question endlessly for years and years and years, and may still be doing so. But actually, it's a really interesting question. Is it the same process? And so I got really intrigued by this. I think he was a little surprised that I must have handed in an essay the size of a PhD thesis discussing why I thought it was or it wasn't the same process. Right? Because actually, when you start looking at the origins of phyla, you discover a remarkable thing. That remarkable thing is that they all date back to one time in Earth's history, or at least all those that we have a fossil record for, and that's the Cambrian explosion. About 540 million years ago, something happened, and at that something happening point, these major body forms appeared. So the fossil record of arthropods and the fossil record of echinoderms and the fossil record of chordates all dates back to this one period. And in fact, no other phyla have evolved since then, as far as we can tell, apart from some of the really weird squishy things that live between sand grains, right? So something really remarkable happened there, which suggests actually maybe it isn't the same process. So I was really intrigued by this idea. And the key book at the time was a book by Stephen Jay Gould, a famous evolutionary biologist, who wrote this book called Wonderful Life about this entire problem. And he suggested that actually at this point there was a remarkable experimentation with body forms. And this is based on these fossils. So these are fossil animals. These are the first animal fossils that we have in the fossil record that we can understand. There are fossils that are in Australia. You can go to the where's that place? Adelaide Museum, and you see them, and they're sort of squishy things. They predate the Cambrian explosion. But after the Cambrian explosion, we get fossils like this in a few places in the world. And these are really interesting fossils. So up in the top corner, I should have brought a pointy thing, shouldn't I? Up in the top corner there, we have a thing which, if you saw it on the beach today, you would know was a polychaete worm. The black thing in the middle with the lots of legs, it's an onychophoran. Those are crawling around the hills of Dunedin as we speak. Right? The one down the bottom in the middle here, This guy here, from the pattern of its muscles and the formation of its head, that's a chordate. That is the ancestor of most of the people in this room. I'm not sure about you. Right? So suddenly in the fossil record, we get these different groups of animals. How did they get there? Why? How did they evolve? And so Stephen Jay Gould's idea was actually there was this and I wanted to say something else before I talked about Stephen. In the corner there's something else, right? Many of these animals are things we recognize, but the one in the corner there has 5 eyes and a trunk, okay? There's nothing like that living on Earth today. Stephen Jay Gould says, this is because there was an experimentation with body forms, right? Animals evolved and they experimented with body forms, and then some of them survived and some of them didn't. It's a really interesting idea, almost probably entirely wrong, but it was a really cool idea. If you take these animals, there are normal things. This is a picture from the Royal Ontario Museum where many of these fossils are, and you can see that actually, there's jellyfish there and there's trilobites, which are strange to us, but actually really common in Earth's history. The big thing up there is a foot-long predator called Anomalocaris, right? There's nothing like that on Earth today. It's a really cool animal, and in fact, I'd like to meet one. Doesn't look much worse than most wolves. So these things are really cool, and they demonstrate that actually there's a lot of diversity in phyla that has disappeared on Earth. And Stephen Jay Gould would say that this is because these animals were experimenting with body patterns. Maybe there wasn't much genetic change which led to these different body patterns. Maybe if you have a developmental system which is sort of modular, you can put arms and legs wherever you like. And that even with very little genetic change, you can get all of this diversity. And when I was reading about things, another set of experiments came out from some people who are even cooler. Ed Lewis, the Nobel Prize winner, whose names I can't remember at this point, and David Hogness. These guys were looking at the development of fruit flies, and they showed some really remarkable things. Things which spoke to me when I was looking at these fossils. In the corner there where it says WT, that's the head of a fruit fly in a scanning electron microscope. In the middle, that fruit fly has got a mutation in it. Would you shut up? A single mutation. And that single mutation, it's not me. That single mutation makes legs grow out where the antenna should be. So the antenna are transformed into legs. The one on the end, both the legs and the mouth parts are transformed. The antenna and the mouth parts are transformed into legs. So single mutations can move body parts around, maybe mix them up. That's kind of similar to what we see, sort of, in that form. And the one down the bottom here is even more interesting. Fruit flies, as you all know, every single one of you know, have 1 pair of wings, which is unusual in insects. Most insects have 2 pairs of wings, right? Fruit flies, or flies, have evolved, their second pair of wings has evolved into a balance organ called a holter. But with a mutation, we can transform it back again. So this fly over here has a mutation which returns 2 pairs of wings to the fly, right? It's like an atavistic mutation which allows this thing to look like an ancestral insect. 1 mutation. Again, you're in this position where you're starting to think maybe we can mess with developmental systems to produce perfectly functioning organisms with wildly different body patterns. The genes that are involved in these mutations are called Hox genes, and they're really interesting genes because they happen in flies, they happen in all of you too. Every animal that we know of, or most animals, apart from those squishy things that you find on the beach, most of them have Hox genes, and they mostly do similar sorts of jobs. And so one of the arguments about what the Burgett tail animals really represented was experimentation with Hox genes, the evolution of Hox genes. Now, all of what I've told you is almost entirely wrong. Right? It's a really good story, but it turns out that these aren't really single mutations, they're really complex. That 4-winged fly does not fly. No matter how far you throw it, it never flies. Right? It's not a functioning organism, it's a mutant. And those mutants tell us something about the development of an organism. They don't necessarily tell us about their evolution. But I was intrigued by this idea that actually maybe we can nail down how much genetic change it requires to have a morphological change in an organism. And so I started to ask these 2 questions in my career. Firstly, how do you make an animal? What do you do to make an animal? And then how do you change the way you make an animal to make another sort of animal, either a differently shaped beetle or a completely different body pattern? How do those processes which make an animal evolve? So, to talk about this, I need to introduce you to one of my favourite friends, Drosophila melanogaster. Hamish is wincing there because he knows it should be called Psophophora melanogaster. Drosophila melanogaster, the common fruit fly, it's called the vinegar fly. We call it that because it's easier to import vinegar flies into New Zealand than fruit flies because the MPI doesn't recognise there might be different sorts of fruit fly. So this is a fruit fly. We've been studying, not me, people have been studying fruit flies since 1910. And it means that we have a whole set of tools to understand how this fly works. We have ways of turning genes off. We have ways of turning genes on. We have ways of turning genes on and off in places where they shouldn't be turned on. And that's remarkably useful and has enabled us to know more about the development of that animal than any other animal on Earth, right? We know how a fruit fly works far better than we know how you work, which is great. So when I started trying to ask the question, how do we make an animal? Obviously, the place to start was in Drosophila. So let me show you how we make an animal. Right, just add water. This is two views of the same embryo. This is a technology, an imaging technology called light sheet microscopy, which Vernon is going to buy after this lecture. It's only $1 million or so. So this is from the Howard Hughes Medical Institute's digital embryo project. We're looking at two faces of an embryo, the top and the bottom, a Drosophila embryo. The embryo is already 3 hours old. Okay? So a lot of the early development's already done. I'm just going to watch the development of this embryo. This is the development from what was a single-celled organism to one with morphology, with structure, with purpose, all right? So I'm going to press the button and stuff's going to happen, and I'm going to talk to you about it. There's two key processes going on here. The first one is called patterning. If you look about, let's see, I've got the timing wrong, crap. Now, you'll see that there are kind of stripes across the embryo, right? Those stripes are segmentation. Fruit flies are insects, they're segmented. That's a patterning process. The embryo, which was undifferentiated, is being divided up into chunks. A process called patterning. The other thing you need to notice is cell fate. You see those big cells up the top there? They're not big cells, they're big nuclei. Those are the cells of a particular tissue called the amnioserosa. They're different from the cells around it because they've worked out where they are through the patterning process and they've changed their fate. They've turned into some other sort of cell. So those two processes are key to development, patterning and cell fate. We're going to talk a little bit about both of them. But I want to show you something remarkable, and it's going to happen about, well, it's going to happen, no, see, I've got the timing terribly wrong. It's going to happen about now. About now. About now. Oh, for God's sake. There we go. Look at that. You see that? It's twitching. Why is it twitching? It just made muscles. It's now learning how to connect those muscles to its nervous system. And in a second, when this film stops, what's going to crawl away is a living, breathing, reacting, working maggot. Okay? It's not quite as cool as if it had been a gazelle, but that's pretty awesome, isn't it? And that was in 18 hours. 18 hours from just laid egg to an organism that works. Patterning and cell fate. It's awesome. So when I started looking at this process, I thought to myself, I want to go and learn how that works. So I went and worked with this guy, Dave Hartley, at the Imperial College in London. And we were studying the development of the nervous system. So the nervous system is obviously a key part of being an animal. There's a picture up there. You see the red picture. It's labelled C. You can see that's the nervous system. That red thing, that kind of comma-shaped object, is the nervous system of a fruit fly. So we're interested in how cells decided to become part of the nervous system, because cells have to decide if they're going to be nervous system cells or if they're going to be skin cells in Drosophila. In the embryo just below that comma one, you can see one where a particular process has been messed up, a process called notch cell signaling. Without that process, all of those cells in the ventral part of the embryo have turned into nervous system cells, right? That's at the expense of skin cells. So that embryo has a big brain and not enough skin. And we all know someone like that, right? Alongside, going over, there's also those whiter embryos. Those are actually the embryos of a completely different organism, a frog. Because it turns out that notch cell signaling, which defines the nervous system in a fruit fly, also defines the nervous system in all of us. Right? It's a very ancient and conserved process by which cells determine whether they're going to become nervous system cells or something else. In humans, when it gets messed up, we die as embryos. Everyone in this room has a perfectly functioning notch cell signaling pathway because you wouldn't be sitting here if you didn't. So I learned a lot about this kind of process and learning how to use Drosophila as a way to ask questions about biology. But then I kind of got the itch to do something different. And so I went back to asking the next question, which is, how do those processes change over evolutionary time? So I worked, shouldn't practice your talk. So that's the process that went wrong, okay? It's a complex cell signaling pathway with a lot of biochemical thingies and proteins and stuff. All of you who are biochemists in the room are saying, oh yeah, that's pretty cool. Everyone else is going, what the hell is that? Remember that there will be an exam on it later. We're coming back, okay? Right. So I went to Cambridge and worked with this guy, Michael Acomb, who worked at the Museum of Zoology at the University of Cambridge. And his interest, Michael Acomb had been a remarkable scientist in studying segmentation, the process that I showed you in that embryo, the dividing up of the embryo into chunks in a fruit fly. He'd done much of the work to understand how segmentation works in a fruit fly. By the time I started working with him, he'd decided that he wanted to work out how that worked in other organisms, and particularly that insect below that, the African plague locust. So I worked on African plague locusts for 3 years, and that's long enough, right? They're not a plague for no reason. They're horrible things. They eat each other, they eat you, they make you sneeze, they poo everywhere. They're just dreadful things. But they are remarkable in that they have a completely different embryo to the fruit fly. They're still an insect, they still get segmented, but there's a completely different embryo. They're also 350 million years diverged from fruit flies, right? That's older than the divergence of us and reptiles probably, and possibly fish. It's a very long evolutionary distance. So by studying what was happening in this species, we hoped to learn new things about how segmentation worked. Unfortunately, the African plague locust is a horrible organism to work with, not only because of its nasty behavior, but also it has a genome which is 10 times the size of the human genome, which means that cloning a gene in the African plague locust is very difficult. So we did, though. And what we found was actually the genes that we cloned, the genes that controlled segmentation in a fruit fly, were the same genes which controlled segmentation in a locust. Right? The gene caudal up there labelled defines the posterior fate in a locust, it defines posterior fate in a fruit fly. Hunchback, the brown stain in the middle of that embryo, defines those central regions in a locust. It defines central and anterior regions in a fruit fly. 350 million years of evolution in a completely different embryo, and we have very, very similar genes. That's all right, we haven't started yet. and we haven't got to bees yet, so you're good. The pearberry and wingless are also genes involved in segmentation in Drosophila. They're also expressed in a pattern suggesting they have a role in segmentation in locusts. Same genes, 350 million years of evolution. But there's details that are different. Some of the genes are expressed in slightly different patterns. The timing by which they appear is slightly different. It's as if these organisms are using the same genes, but they're using them slightly differently. After working there, I went to Agriculture Canada, which was an eye-opener, and worked with this thing, the two-spotted spider mite, right, which is a pest of agricultural stuff. It's also not an insect. It's more closely related to spiders, and the common ancestor between spiders and insects lived before the time of the Cambrian explosion, right? In the Burgess Shale, there are the ancestors of insects, and they are the ancestors of insects, and there are the ancestors of spiders. So this is over 540 million years diverged from Drosophila. It uses the same genes to segment its body, which is weird. It turns out when we sequence the human genome that many of the genes which segment the Drosophila embryo are also in your genome, suggesting that actually the genes that are used in development are very highly conserved. They don't change very much, which raises the question, how do we get any change at all? If we're using the same genes, why am I not a beetle? So it's very difficult to lecture in this thing because it keeps sliding off. So if I do I'm actually thinking it would make good beachwear, apart from being flammable, right? It would be awesome. And I think if you took it in the sea, you'd drown, right? Disappointing. So what we were doing was depressing to me. At the end of those two postdocs, I thought it was one of those moments where, as a scientist, I'm sure we've all had them, it's like, is it time to give up science and take up knitting for a living? Because frankly, this is going nowhere. We're defining that the same genes do these processes, but we don't understand what those genes do or how they do it. We can't even prove that these genes are actually involved in the processes we think they are, right? Because we can't do the key experiment, which we can do in Drosophila, which is to turn them off and see what happens. We can't do that. Why are we bothering? And also, how do I understand the way a gene changes its regulation when I don't have any information? I don't have the genome sequence of a locust. So how do I work out how those genes are changing the way they work? And so I was really kind of depressed about this. But the good thing is that in that depression, I turned to the internet and discovered a very salient fact which made me change the way I worked on this subject. So when I started being a PhD student, I was offered 2 PhD places, one in London with Dave Hartley and one in Cambridge, which was to sequence the alcohol dehydrogenase locus of the common fruit fly, Drosophila melanogaster. Right, which one would you take? London or boring stuff with drug-related things and flies? Well, I got it wrong, because that project was the start of the Drosophila genome project. On the way to sequencing the human genome, as practice, they sequenced the genome of the fruit fly, and that changed the way we work. Suddenly, all those guys who worked in fruit flies had enormous resources that I never had. So I wanted that. That was good. That would help me understand how genes change their regulation. And the next thing that happened is they sequenced the human genome. Good, excellent. Don't really care about humans, but good. The key thing is they got it out of the way, and some universities and research institutes had spent millions of dollars on sequencing equipment, and they'd done the genome. What the hell are they going to do now? Well, so what do? You start sequencing things you think are important. The next insects to be sequenced were mosquitoes. And I thought to myself, I could work on mosquitoes. And then I thought, no, I don't really want to work on an organism that sucks my blood. That would be a mistake. Also, difficult to import to New Zealand because they carry malaria and dengue and other things. And then I heard they're sequencing the genome of the honeybee, a species I really didn't care anything about. But the honeybee turned out to be the solution to all these problems. So honeybees are really important insects, and you all know that, and we probably won't talk about it. They're the most, I like to say they're the most economically important insects, but that's probably a lie. They're certainly important for most human food production, and they're nice and friendly, and we like them. They're good things, okay? And that's why their genome was being sequenced. And sequencing the genome, we started to learn about, looking after bees. So when I came back to New Zealand, I was really pleased to be hired by the biochemistry department working on bees. I still don't believe that they realised what the hell I was supposed to be doing, though Catherine says that they did. And I'd really like to thank Warren for taking a punt. Frankly, there was a guy going to work on bees, and he hired me. And I'd also like to thank Clive. I don't know if Clive Ronson is here, but he also let this idiot work in the genetics teaching program when my genetics is pretty poor, frankly. So thanks, Clive. Honeybees are awesome. Having the genome sequence of honeybees was really good. When I heard that they were sequencing the genome of the honeybee, I started doing some work in honeybees, published some papers, and got involved with the honeybee genome sequencing project. I got asked to be involved, and so my lab and the lab of Alison Mercer, also at Otago, helped publish the honeybee genome sequence in 2006, which was a really key achievement for us. It taught me a whole load of things. We did all kinds of things that I'd never done before. But it also helped us identify all of those genes that we thought were involved in sequencing and segmentation in Drosophila. We could see them in the honeybee genome as well. We could look at those genes and work out how they were regulated. So now we had a way to look at the way gene regulation changes. We still needed a technology to turn genes on and off. And luckily, at exactly the same time, that technology came along. It's a technology called RNA interference, and it's complicated, but it's a process that occurs in all eukaryotic cells. It turns out that we respond to double-stranded RNA. If we put double-stranded RNA of a particular sequence into a cell, then those cells respond by taking that double-stranded RNA, reading the sequence, and using that sequence to destroy either RNA of a similar sequence or to turn off the DNA copy of a gene. So it enables us, if we have the sequence of a gene, to turn that gene off. Okay? To get double-stranded RNA into a honeybee embryo is tricky. It involves micro-injection, and the picture you can see up there is a single honeybee embryo, and the sort of green thing coming in from the side is a needle filled with double-stranded RNA. That needle is finer than a human hair, and we're using it to inject that embryo to turn off a particular gene. With this technology, we could now turn off individual genes during segmentation in the honeybee and ask, did these genes do the same job as they did in Drosophila? This work was funded by the Marsden Fund and by a fantastic postdoc, Megan Wilson, who showed me this result and I almost fell off a chair. Here we have 2 larvae. Honeybees also produce maggoty-looking things. The one here that says DS-EGFP is a wild-type larva which has been injected with a piece of double-stranded RNA which isn't present in the honeybee genome. That's our control. The one next to it has been injected with double-stranded RNA against a gene called giant. And giant, in Drosophila, controls the development of the head. And in honeybees, when you knock it out, the head disappears. It says that actually it's doing the same job. So that enables us to test the function of a gene. We can also, using the genome sequence, look to see how those genes are regulated. In Drosophila, at the top there, euphemistically called drossies, we have a picture of the giant gene, right, in the genome, and those little taggy things sticking off the genome sequence, those are places where proteins bind to that DNA, all done by other people using Drosophila's genetic tools. Those little regions which are in boxes, those are regulatory regions. These drive the expression of giant. And the embryos underneath it are all multicolored. You only need to look at the green color. That's where giant is expressed in the Drosophila embryo. Down the bottom, we have the same picture in bees. We've got the embryos down the bottom. Giant is expressed in anterior regions, just as it is in Drosophila. We can also identify regions of the genome just upstream of the giant gene in honeybees, which we think regulate it. We can identify the proteins involved in that process, the proteins that are binding to that DNA and are turning the giant gene on and off. And we can test those predictions with RNA interference. This is going to be very difficult to do without a pointer. So at the top here we have an embryo which is all blue. It's stained for a gene called caudal, AMCAD it says there. That blue staining shows where the RNA from that gene is, where that gene is turned on. Okay, underneath it there's a gene called hunchback, which is expressed in the anterior regions of the embryo. Underneath that one called orthodentical. Okay? These are the proteins, well these are the RNA, the proteins, these are the genes which encode the proteins which bind to that sequence of DNA upstream of the giant gene. So when we knock them out, if those proteins regulate the giant gene, then they should change the expression of that gene. And that's exactly what we see. So over on the other side, there's an embryo which is all blue. That's an embryo that is stained for giant, which at the bottom you can see is expressed in anterior regions of the embryo. When we knock out caudal, it's expressed everywhere. So caudal represses giant. All right? The next embryo down, we've knocked out hunchback. Hunchback must activate giant because when we knock out hunchback, there's no giant. The same with orthodentical and another orthodentical gene we don't like to talk about underneath. All of those genes and those interactions allow us to draw network diagrams of how these genes work together to segment the embryo. And we know what that network looks like in Drosophila. It looks like that. 30 years of research on Drosophila has indicated that those are the network interactions which segment a Drosophila embryo. All right? Don't try and understand it. It's inexplicable. It can only be understood by modeling in a computer, and most of the time when people try that, it doesn't work, which indicates that while we think we know what we're doing here, we don't really. When we test those interactions in a bee, what we see is they're different. They're a different set of interactions. Same genes, different set of interactions. So what's happening during the evolution of this process, in that 250 million years of evolution between bees and flies, is not new genes evolving. It's the genes remain the same, but the wiring diagram that holds them together changes. Right? Suddenly, there's a lot of developmental evolution going on here, but there's not a lot of actual change. When we look at segmentation in a bee and segmentation in a fruit fly, it looks almost identical. But underlying that almost identical nature, there's a hell of a lot of evolution going on. So while I went out looking to see if I could find cases where a small amount of genetic change led to a large morphological change, what I found was a case where not a lot of morphological change was underpinned by lots and lots of evolution, which annoyed me immensely. And was another one of those moments when you say, actually knitting looks like a better occupation than this. But then I thought, actually, Peter, you are one of the biggest idiots that has ever walked the earth, because bees already tell us the answer to the question I wanted to know. Bees already produce vastly different morphologies with no genetic change at all. You all know that. I'd forgotten. Bees produce 2 female castes, workers and queen bees. So there's a honeybee looks like a honeybee, but they will produce drones, which are male bees and are so embarrassing we're not going to talk about them. And they produce workers and queens. Workers and queen bees are both female bees. Right? We can induce any worker, any female embryo, any female larva we like to form a queen just by changing its position in the hive. And queen bees are larger, they lay eggs, they behave differently, they smell differently, they are different colours, and they live 20 times longer than worker bees. Right? These are not minor changes. This is a very different organism. And that very different organism is triggered with no genetic change at all, just diet. So I came to the revelation that actually I've been doing these experiments wrong all these years. Instead of studying processes where things don't evolve, things don't change, and understand what the developmental processes are, why are we not just studying this? How do we get queen and worker bees How does diet trigger these kinds of changes? Now, I would tell you about this, but when I started giving this, practicing this talk this afternoon, it was an hour and a half long. And so I've been throwing out slides all afternoon, and I accidentally threw out all the slides involved in this process. I do apologize. It was awesome work. Really fantastic stuff. But actually, the problem with it is that it's really hard to test what we find. We can find genes which are always turned on in queens, which we think are required to make a queen bee. But testing that and trying to knock out those genes in a bee, and seeing if we can produce queens or not, is really, really hard. It involves feeding larval bees in the lab, and there just are not enough graduate students in the world prepared to stay up and feed bees continuously. Okay, so we gave that up. We did it, we published it, we gave it up. I want to talk about another thing that bees do, which is kind of similar. So it's talking about a change, an environmental change, which triggers different development, different morphology. And it turns out that actually there's another thing that bees do related to caste development, which is the same kind of process. Queen bees repress worker bee reproduction. You know that because in a hive, you all know the queen bee lays all the eggs while the workers do all the work. Okay? But actually, the queen actively represses reproduction in their workers, and they do it through a pheromone called queen mandibular pheromone. So if we look at the ovaries of a worker and a queen, the ovaries of a queen bee, under the queen there, are huge. Actually, those are some of the biggest ovaries you'll see in the insect world. You should be impressed, but you're obviously not. In a worker, they're much smaller, and that's to do with that royal jelly diet that they get early in life. Okay? It's not terribly exciting. But the key point is that if you take the queen away from a hive, those worker ovaries activate. They start developing and producing eggs. Okay? And it's a process like a change in form. And so I thought, actually, here's a model system that I can work with, because I can manipulate this with all kinds of nasty ways. This is a process that we could work out how this works and manipulate it. So we wanted to ask the question, how do worker bees respond to queen mandibular pheromone, and what happens when you take that queen mandibular pheromone away? And luckily, the third technology which really changed my research came along. We sequenced the genome, that changed everything I did. RNA interference changed everything I did. And the last thing was high-throughput genomics. Oh, it sounds rock and roll. So once people had sequenced the human genome, they turned their attention to finding cheaper ways to sequence DNA. Cheaper, faster, better. It cost several $1 billion to sequence the first human genome. We can do it in the biochemistry department for a couple of $1,000s in a week's worth of your time. Okay? This has changed because of the way of fantastic achievements in microfabrication and ways of dealing with massive amounts of data. This means that actually we no longer deal with individual genes. Gone are the days where I look for the hunchback gene. Now I look at loads of genes, all the genes in the genome, and how they respond in a developmental situation. So I'm going to do some teaching here, right? Warren is going to throw something at me about halfway through here, so you'll look out for this. I want to tell you a piece of information which is the central dogma of molecular biology. Do I need to stand further away? Okay, so it's called the central dogma because molecular biologists don't believe in law, right? There's no law of thermodynamics in molecular biology. There's just the central dogma. It's called the central dogma because, like most dogma, it's wrong in all kinds of ways. But the idea is that information flows in a cell from DNA to RNA, another sort of molecule, to protein, okay? And the proteins do stuff. This is where Warren throws something at me because he knows very well that actually RNA does stuff as well. But we're not going to talk about that. Protein does stuff, and that information comes from DNA. And RNA is the molecule in between that carries that information. I like to think of the genome, stupidly, as a medieval library. And follow me on this, it's a really good metaphor. Right, in a medieval library, you've got all these big books with the information in them, but because they're so valuable, you chain them to the shelves. Right? You can't take the books out. So Bob the baker, who wants a new recipe for whatever they eat in medieval times, he goes to his books. He can't take the book out to his bakery, he has to copy it out. Right? He copies out the recipe and he takes it off and turns it into protein, which does stuff. But he's not an idiot. He doesn't copy the whole book, he only copies the bits that he needs. And cells are like that as well. They don't to make RNA from the whole genome, they make it from only the genes that they need. And so if we look at the molecules which are in RNA in a cell, that's a reflection of what genes are turned on. Okay? So we can take an active ovary, one where the queen bee has gone away, and sequence all the RNA, and those are the genes which are required to make an active ovary. And if we take a repressed ovary, we can look at all those genes and compare them and say, what's different? What's changed across the whole genome? Which is remarkable, right? Using high-throughput genomics, we can sequence every piece of RNA and say which genes are on in this tissue that are not on in that tissue. So, working with a bunch of people, with the help of Sir Peter Gluckman, who's the guy with the hamster beard there, and Hugh Blair from Massey University and Frank Bloomfield from Auckland University, we undertook to look at differences in gene expression between active workers, repressed workers, and queen bees. And all this work was done by Elizabeth Duncan, who is now at the University of Leeds, right? Much better university. No, kidding. So we took this RNA, we sequenced it, and we looked at what genes were different, what changes between these two conditions. And the really frightening thing is that a moment in my life where I really thought, actually, everything I've done isn't completely pointless, we found this. You've seen it before, it's notch cell signaling, right? It was just amazing. I was looking at lists of gene names and going, I know all these names. These are genes I've worked with in Drosophila. It's notch cell signaling. My God, it's notch cell signaling. What does notch cell signaling do? It controls the development of the nervous system. And here it is. It's the thing that's changing between active and inactive worker ovaries. Now, let's get biochemical for a moment. Notch cell signaling is a process whereby two cells talk to each other. Without a pointer, this is going to be fun. One cell up the end there, you can see the cell in the top corner, it's talking to the big cell down the bottom here. It's talking through two proteins called serrate and delta, and they bind to that big chunky thing in the middle called the notch receptor. When those proteins interact, a bit of the notch receptor is cut off by that lump of things that says gamma secretase complex. All right? And that bit of the notch receptor goes into the nucleus where it turns on some genes. Turns on some genes called the enhancer of split complex, the world's most stupid name for a set of genes. Okay? We're going to remember those things. Remember, a bit of the notch receptor goes into the nucleus. When signaling is occurring, a bit of the notch receptor is in the nucleus. It's turning on a set of genes, the enhancer of split genes. Genes that, lo and behold, are studied in the honeybee for their role in nervous system development. Right? There's a complex of genes. They're so cool. I'm aching to tell you about it, but I can't. Right? Come and talk to me after. Say, tell me about the enhancer of split complex. Awesome. These genes respond to notch cell signaling. So in a cell which is receiving a signal through that notch cell signaling pathway, these genes are on, they're turned on, they're expressed, okay? So we can identify every cell which is receiving a signal through that notch receptor because it will express enhancer of split genes. So we said to ourselves, where in the ovary is this role of notch cell signaling happening? And we found it in one place. These blobby diagrams down in the back there with a blue staining, those are staining the nuclei of cells of the germarium of the ovary of a honeybee. Okay? This is just a part of the ovary, it doesn't really matter. It's the bit that really does stuff because in the germarium are the germ stem cells, which are the cells which go to make oocytes, right? So in that region, what we've got here is that's the staining in blue, those are the nuclei. On the other side, those are the same ovaries, but stained for enhancer of split genes. And those are actually ovaries from different bees. So in the top, that's an ovary from a queen right worker, a worker with a queen in the hive. Underneath, that's a worker without a queen. And at the bottom, that's a queen ovary. And the enhancer of split gene is only turned on in the top one. When a queen is in the hive, the enhancer of split genes are turned on in the germarium of the ovary. Okay? That's the only time I've ever found something so clear that it stands out like that. Kind of, wow, that's amazing. Every time I look at this diagram, I wonder if I've made it up. It's so clear. It's on where you have a queen. Take the queen away, it's off again. I thought that was a good point to interrupt. Good work. We can show that this is actually due to notch cell signaling because, as I told you, a bit of the notch receptor, when signaling occurring, goes into the nucleus. Okay? If we tag that bit of the notch receptor with an antibody, if notch cell signaling is active, we should see it in the nucleus, right? So under there, in the red staining, under repressed, you can see those little blobby things. That's, again, the germarium of an ovary, and those little blobby things are nuclei. I know they're nuclei because just underneath the red staining, there's blue staining, which is staining DNA, which lives in the nucleus. You know that they overlap because if you look right at the bottom and ignore the green stuff, it's purple. The red and the green overlap entirely. The notch receptor is in the nucleus, notch signaling is active. So in a repressed worker, notch signaling is active. In an active worker, there's no notch receptor, it's gone. We can't see it at all. So there's a clear difference. The notch is signaling going on when the queen is there, and it's not when the queen isn't there. But here's the problem, right? What I've shown you is the same old crap I keep showing you. It's correlations. It's, oh yeah, these gene expressions look like they do the thing. How do we prove it? Well, we prove it by doing manipulative experiments. And that's why it's fun to work on bees. We can't do RNAi in adult bees because they don't like it, but we discovered a way to block notch cell signaling in another way. And the way we did it was using a drug. So I told you that the notch receptor is cleaved by a little complex of proteins called the gamma secretase complex. And those of you who are Warren will be going, the gamma secretase complex, that's the thing that has a role in Alzheimer's disease in humans. It's a drug target for Alzheimer's disease. Bees don't get Alzheimer's disease. They use their gamma secretase complex to cleave the notch receptor. But there are lots of pharmaceutical companies out here who have developed drugs to block the gamma secretase complex because they think it might be therapeutic for Alzheimer's disease. So we decided to put our bees on Alzheimer's drugs. Doesn't make them forget things, but it does something remarkable. So the drug we're using here is a thing called DAPT. It's got a real name, but I can't pronounce it, and it's several miles long. So we're going to call it DAPT. What we did is we took bees and we shoved them in that little box up there. All right, so we took about 50 to 100 bees and we shoved them in that little box, and we fed them either DAPT or food without DAPT. We fed them all food, but food with or without DAPT. Okay. The box is a really highly technical thing. It's closed with a piece of tape. So I'd like to thank Otto, who volunteered his thumb to stick over the hole while we were changing the food. That's good. Something I would never have done myself. I don't know if Otto's here, probably because he's still in hospital. Bees shoved in a cage without a queen start laying eggs, okay? Because they activate their ovaries. We can also buy queen mandibular pheromone because actually some Canadian chemists discovered all the components of queen mandibular pheromone, and it's used by beekeepers to look after their bees. So we can treat these bees with queen mandibular pheromone, we can treat them without queen mandibular pheromone, and we can treat them with or without DAPT. And that's exactly what we did. What would we expect? If notch cell signaling is required to repress the ovaries, when we block notch cell signaling, they should activate their ovaries. That process is downstream of queen mandibular pheromone. Right? So queen mandibular pheromone should lose in a fight to DAPT. So what we did is we took the bees, we ripped out their ovaries 2 weeks later, right? And we scored them double blind, fantastic experiment, on the scale of ovary activation from 0, which is not active, to 3, which is really active. And we used those numbers to provide an ovary activation score, and we looked at how that ovary activation scored with our various treatments. Bees without QMP, which says control minus QMP, those guys activate their ovaries, as you would expect. When we give them QMP, which is the middle thing, control plus QMP, they don't activate their ovaries. Okay, their ovary activation score is lower. But if we give them DAPT and QMP, they activate their ovaries more. There's 2 copies of this experiment because it turns out there's also an input from season. One of those experiments was done in spring and one of them was done in autumn. In autumn, bees love to activate their ovaries, and that's the second experiment. You can see that actually with DAPT, they activate their ovaries just slightly more than if they have no QMP at all. DAPT beats QMP. So we've shown that notch cell signaling, this ancient conserved process which controls nervous system development, has been taken by queen bees to control workers. Right? Here's a process that has evolved. It's evolved in bees. This is a bee-specific thing. What did they choose? Did they evolve new genes? Did they even invent some genes that I didn't know about? No, they went and picked genes that I'd studied as a PhD student. Classic developmental genes. Right? There's no new genes here. It's just fiddling with the ones that we actually have. So what have I learned from this process of studying developmental processes? Well, I've learnt that actually the process that I naively thought could be fiddled with easily to produce different morphologies is actually incredibly complicated. That these processes are rewired constantly during evolution, and it's that rewiring of conserved genes and pathways that's important. Even when new things evolve, it's often old genes that do those processes. We don't invent new genes during evolution, we just use the ones we've got. Also, I like to think that developmental evolution is a bit like a swan on a fast-flowing river, right? It looks like nothing's really happening, but even when developmental processes are the same, such as segmentation, the little feet of that swan are working damn hard, rewiring those networks over and over again. And I'm beginning to believe that actually those networks might drift, that actually if there isn't strong selective pressure on them, they might change just for fun. As long as they work, those networks might be really easy to change. Turns out that there's no simple answer to these problems. I thought there was going to be a simple answer, and it turns out there isn't. And actually, I'm more enthused than I ever was because that complexity is really interesting. And now, ladies and gentlemen, just because Vernon said so, we use this stuff. I've been talking to you, rabbiting on for what seems like several hours about fundamental developmental processes. And I'm really glad that none of you seem to be only a few of you seem to be asleep. The real kicker here is that actually non-model insects, apart from Drosophila, we know nothing about most insects. Yet the most diverse group of animals on Earth are beetles. The second most diverse group of animals on Earth are mites. Bees, ants, aphids, insects are really, really important, and we know very little about them, and yet they're really important to us too. Bees, their pollination is required for our food production, and so apparently a third of the crops we eat require bees for pollination. A third of the stored grain we collect is eaten by beetles, right? What is our response to this? Well, we think all insects are pests. Most of you in this room think they're icky, and I bet most of you have a can of black flag somewhere in the back of your drawer, right? Which you've sprayed, which is a compound which kills every insect and spiders and nematode worms and most other things on planets. We have this really vertebrate view of the world, and we've got to get over it because actually insects are awesome, right? So we're trying to use the technologies and the ideas that we've developed in these fundamental projects to help solve problems with insect biology. For the Ministry of Business, Innovation and Employment, we're developing selective insecticides. By understanding the genome and the development of bees, we can target pathways which work differently in bees to other insects and hopefully find chemicals which kill other insects, leaving our bees alone. For the Bioprotection Corps, we're using those same technologies to address some of the pasture pests in New Zealand, particularly the Argentine stem weevil. For the Biological Heritage National Science Challenge, we're deploying RNA interference we developed in bees to kill wasps. I don't think it's a very good idea, but we'll do it anyway. And for the Ministry of Primary Industries and for the AgMart, we've been studying inbreeding in New Zealand bees to try and make sure that we have healthy bee populations in this country. None of this is an extra bit of research. None of this is, oh, that's applied research. It's not as good as this stuff over here, which is your fundamental research. It's all a continuum. And I'm delighted that actually I developed technologies to turn off genes in bees, but I can use that to turn off genes in a wasp and perhaps kill them in new and exciting ways, or at least specific ways. Alongside this, everyone's mentioned it, so I'll just skip through it. Science doesn't happen in a vacuum. We need to talk to the taxpayers who pay for our research. We need to talk to the communities who are going to implement and use our research in future years. And so I'm really proud to have been involved with the Lab in a Box project, which to date, and Craig is going to throw something at me at this point, has interacted with almost 10,000 rural New Zealanders and talking to them about all kinds of science, from physics, chemistry, marine science, but mainly about insects, particularly bees. I think that's right. Lab in a Box is a collaboration with lots of people, including the Otago Museum. So I've been really delighted that actually my research can be translated into things which are useful for industry, things which are useful for the environment, but also can get out to those people who are going to use those kinds of technologies. And I urge you all to do the same thing. Finally, they say that it takes a village to raise a child. I don't really know what that means because I don't live in a village and my children seem vaguely okay. But it certainly takes a village to raise a scientist. And so I'd like to thank all of these people. And I hope you're reading that and seeing if your name is there. If it isn't there, that's probably an oversight because you've all helped me so much. There are people who have worked in my lab in there. And to those people, I'd like to say, sorry. There are people who are my collaborators, who are people who have helped me get involved in all kinds of new ideas and things. And so, you guys have been awesome. Thank you so very much and stuff. And yeah, you all deserve to buy yourself a drink and drink it. Finally, of course, the people I need to thank the most, this lot, not the guy in the middle with the stupid shirt, but my family and my fantastic wife. Science is an immensely dispiriting occupation, right? And I say this to all of you young scientists aspiring to my job. It's really depressing. I submitted a paper to Nature recently and noticed that I'd had 6 papers rejected by Nature in my career. And that's kind of one a year. It's really quite depressing. And it's nice to know that even though your colleagues and the scientific community think your work is rubbish, your family still like you. It's nice to know people who judge you not on your scientific output. And so I'd like to thank my family for providing sanity in this difficult business. That's all from me. Thank you so much for listening. It's been a privilege to work with you and to talk to you tonight.
Tonight, because following behind Peter is really not something that anyone wants to do, to have to speak behind Peter. I think he's really just, I guess, provided us with a fine description of his research and his passion and inspiration. I think if you've missed that, I think no one in this room will have missed that, because he is one of the most passionate researchers and communicators that we know. And so it's really a privilege for me to speak and to thank Peter firstly for his talk. I think he's given us a beautiful tour through his research and from his career, which is from Wellington to London, on to Cambridge, and then back to London, but in Ontario, and then on to Dearden. I think for the young scientists here, his talking about the decision-making processes and the early thoughts that he had and the way he changed and then sometimes came back around is really a lesson for us all, and it was a beautiful description of how sometimes things come back around. He's, I think, far from me to describe what he's talked to us about, because he's really communicated very eloquently to us, but it's really about taking these snapshots and thinking very clearly about what he's doing. And I think, to me, Peter's real sort of the magic that he has is the ability to communicate and to put his research within the bigger context, and not just to many of us who are biochemists think about pathways and A binds B, activates C, and I know I've stood here and given a talk like that, and far more of you went to sleep in that talk. And so it's not that is not the good things, that is also not a sort of an important thing to discover, but this ability that Peter has to see the greater vision and connect his research. I think it comes through both in his communication and his grant writing and his paper writing. And this afternoon, when I was looking up a few things to say, I got out his recent paper in Nature Communications and read through some of that. It's really a very nice introduction to a field that I don't know about, and the discussion puts the work nicely in context. And I think you will all, if you look at, goes right back and references Darwin's work, and then some papers from the 60s and 70s. So really describing how sort of the scientific story builds on previous literature, and it's really a very great skill. And I think Peter has won numerous awards for his communication, and I think you will all see why these have been given. So I think we've really seen a snapshot of Peter's research, but I really just want to comment that the biochemistry department is very proud to count him amongst our number. He's a very inspirational person to have on the staff, and always with ideas, and we can see tonight he's got a list of things that Vernon needs to buy him, or maybe Harlene's budget will have to come in for some of these. He's never without a thought or a challenge, thinking about the next step. And so this is a great thing to have in a person. Sometimes a little sort of directing in the right direction is required, but it's always great to have new ideas. A university wouldn't be a university without someone pushing the boundaries and thinking about the new technologies and how we might apply that. And actually that image of that embryo developing was super cool. So I really think Vernon should buy Peter one of those. And and I think Harlene should pay the depreciation, because I don't want the depreciation on my budget. Okay, so really, I think that covers most of my what I wanted to say. Oh, I also wanted to thank Warren, and Peter's partly touched on what I was going to say, but this afternoon I got out Peter's application. So Biochemistry has a very good filing system. I think Tina's here, and I found Peter's application for his job in 2002. And so Warren, it was the HOD then, and appointed Peters to his position. And Peter wrote a very nice included in his application his research statement about what he was going to do. And I have to say, he's pretty much doing exactly what he said. So although he says he's not really a biochemist, he didn't tell any lies in his application. He said he was going to work on model systems and look at evolution by looking at developmental pathways. He said he was going, in his application, he was going to apply that to, in different ways in terms of improving the environment and managing pests and risks. And so all of that is written in his original application. So I think he's been pretty true to form and delivered, I guess, far more than you might hope for. So fabulous, Peter. So I guess tonight I congratulate you, and it's my privilege to give you a small token.
Thank you.
So thank you very much. Awesome.